talk. And yeah, we have already listened to very interesting talks. Now it's my turn, and I'm going to talk about horizontal gene transfer and the Fitch relation. And I'd like to put this work, before I go into the details and talk about the math behind, a bit in some context. And let us for a moment assume that we know the true evolutionary history. And in case we do not have hybridizations, then the history of the species is in general a tree. So in this talk, we are going to talk about rooted trees, phylogenetic trees, and one of these species trees is drawn here. Now, when we talk about species, we essentially talk about their genomes, which are subdivided into small little parts called genes. And also these genes evolve along the rooted species trees in a tree-like fashion. And you can see here some individual genes of the ancestor species, and it evolves along the species tree in this way. And you can see that there are several colors and arcs on this gene tree. And these things reflect the mechanisms that act on the genes through evolution. So we have here these blue square, for example, that reflect duplication events. That is a particular gene gets duplicated, maybe it gets a new function or it gets lost at a certain point. We have speciation events where the entire species, where the species subdivides, so to say, into new species, and the offspring species inherit the entire genome. And we have horizontal gene transfer, and this is a particular transfer of genes across distinct species. So these are the basic events, and based on the true history, we can define several types of homology relations. And homology relations are binary relation on genes that have a common ancestor. So one of these relations have been discussed in one of the previous talks. These are best matches that reflect if genes are closest relatives with respect to the species in which they reside. We have a well-known relation that is the orthology relation. This reflects what their two genes are in this relation whenever the last common ancestor is a speciation event, so it is a red bullet in the gene tree here. And we have the xenology relation. This is defined in terms of horizontal gene transfer. I will make this later more precise. So there are several types of ways on define a relation on homologous genes given the true history. But there's one important thing. The first point is that homology relations have many applications. So they are used in comparative genomics, genome annotation, or just to understand the mechanisms that act on the genes. The other point is that homology relations have a precise mathematical definition in terms of the true history, which we don't know, okay? So there is a well-defined mathematical definition that defines these relations, and this is based on the true history. Now, there is one small little problem. We don't know the true history, right? But these relations are of importance. So there are many methods that allow to infer these relations without any knowledge on the gene or the species trees. There are methods that use sequence similarities, sequence alignments like BLAST. They give us essentially what we know as the reciprocal best match relation, or at least an approximation of it. And based on these reciprocal best matches, one can then start to infer the orthology relation or the xenology relation, and so on. Now, we get only an estimate of the truth, right? And it is quite likely that there are measurement errors in there, that there's noise in there, that this does not make any biological sense. But how can we know it? Because we don't know the truth, right? And so, I mean, to some extent, we want to correct these estimates to some biologically feasible one. But to this end, we need to understand the mathematical structure of these relations. And it is somehow a big surprise that also there have been many methods established to infer these relations, the mathematical structure of these relations have been somehow unknown for a long time. And this is where I worked in the last years heavily on to understand all these type of different relations and try to get an idea on characterization and also on the complexity of editing processes and so on. So, and today I'm going to talk about this particular xenology relation, so a relation that is defined in terms of horizontal gene transfer. Before I start with the mathematics, I'd like to give you a few nice examples that show that horizontal gene transfer is not something weird, it's something that really happened. So, I mean, the classical example is the transfer between bacteria, where you see here these two little bacteria, and one has this arm, spreads it out, and then along this arm, some genetic material is transmitted. And horizontal gene transfer is a significant cause in increased drug resistance when one bacteria cell acquires a resistance and the resistant genes are then transferred to other species. But there are other examples, like from bacteria to animals. So a classical example is, for example, the coffee berry borer beetle. So this little guy is really a major pest because it destroys the coffee. So it has a particular gene that allows them to break down the complex sugars in the coffee beans. And there are clear hints that this is a gene that is a transfer gene that comes from the beetle's gut bacteria. And yeah, so this is another example. But there are really strange examples like horizontal gene transfer between plants. So one of these examples is ferns. So ferns have a particular gene which is called neochrome gene. And this gene allows them to produce very unconventional photoreceptors that can sense both blue and red light. And this is an advantage in low light environments. And this particular gene is not part of any higher plant, and it was long unknown how this particular gene have been evolved, so to say. And there is a strong evidence that the ferns acquired the neochrome gene from this moss-like plant, the hornwort. There are plenty of other interesting examples like horizontal gene transfer from fungi to animals, the beteloid rotifers that have around 8% of their genes from bacterial origins, or even humans are supposed to have more than 100 genes from other organisms. So this should serve somehow as motivation to look at genes and horizontal gene transfer, and in particular the mathematics behind. Back to our true history, there was one very famous paper by Walter M. Fitch. There was a long discussion. It's really interesting to see the papers and the citations in these papers on how to call particular genes and how to call or give them the terms on how they are related. So, and one of the most commonly used definition goes back to Walter M. Fitch, which says that two genes X and Y are xenologues if their history, since their common ancestor, involves an interspecies horizontal transfer of the genetic material for at least one of them. It's a nice definition, right? So let's have a look to this tree here. So this is just a gene tree. We do not need to consider here the underlying species tree. And we have these particular types of edges, of colored edges and vertices. So, and this definition of Walter M. Fitch can simply be rephrased that is on the path from X to Y in the gene tree T, there is a transfer edge. This is simply the translation of this long sentence. Right, so by way of example, 2 and 3, they are xenologues because on their paths there is this transfer edge, while 2 and 5 are not because on their paths in this tree there is no transfer edge. So horizontal gene transfer is a directional event, right? So we know that there is a clear distinction between the horizontal transferred copy and the original that continues to be vertically transferred. And before I go to this, say, symmetric case, I look here at the refinement. And this is the following. What we say in the following is that a gene X is xenologous to a gene Y. In symbol, X points to Y. If on the path from the last common ancestor from X and Y to Y, there is a transfer edge. So you can see here that the different type of or examples what may occur. So we look at two genes. We look at their last common ancestor. And if on the path from X to Y, on the last common ancestor from X to Y to X, there is a transfer edge, but not here, we would have Y points to X. If we have a transfer edge on the path to Y, but not on the path to X, then X points to Y. We may have transfer edges on both sides here of the path. Then we have these bidirectional edges, or we simply have no transfer edge at all, and then we don't have any edges between X and Y. And you can see here the relation that is defined on this tree. The important thing is, I mean, this relation does not depend on any colorings on the vertices. It just depends on whether there is a transfer edge or not. That means we can simply look at edge-labeled trees where we have, yes, this is a transfer edge, or no, this is no transfer edge. It's a binary label on a binary edge label on the trees. So, and this is what we call a Fitch tree. And a Fitch tree is simply a rooted phylogenetic tree together with a binary edge labeling lambda saying this is a transfer edge or not. Again, this is a definition. We say two genes are xenologs if on the path from the last common ancestor of them to Y, there is a transfer edge. Then X is xenologous to Y. And a Fitch relation contains all the pairs of xenologs that I can obtain from a Fitch tree, right? So X of T lambda comprises all pairs of xenologous genes that I can get from this tree. So what we can see here is, say, this is a true scenario. And we have here the corresponding Fitch relation that is all the vertices, I mean, yeah, point to X because on the last common ancestor to this path to X, there is this particular transfer edge, while on the remaining passes, there is simply no transfer edge. This is a simplified Fitch tree with a 0-1 labeling, transfer, no transfer edge. And you can see here another Fitch tree that explains the same Fitch relation. So this already shows that the trees that explain a particular given Fitch relation don't need to be unique. Now, what are now the central questions? The first question is, I give you an arbitrary relation that I have obtained from a Fitch tree. The question is, can we reconstruct the tree, right? The second question is, can we characterize these relations? So now I give you an arbitrary binary relation and ask you, is this a Fitch relation? Is there a tree T lambda such that R corresponds to the particular Fitch relation that I can obtain from this tree? And these are the two questions I'd like to discuss here. So the first question refers to the phylogenetic signal. I give you a relation that I have from some tree, and now I'd like to know how much information about the tree is contained in this relation. Short intermezzo, I know that many of you know what root triples are, but just to ensure that we are talking about the same things, we have here a rooted phylogenetic tree, and we say that a tree T displays a triple AB set if the path from A to B does not intersect the path from set to the root. So the path from A to B does not intersect the path from set to the root, and then we say that this tree displays this triple. And of course, there are many triples displayed by such a rooted phylogenetic tree. We have a triple ABX, for example, or ABY or ABZ, but also all the combinations XYA and so on. There is a well-known algorithm that is called build, and this returns a tree for a given triple set that I derived from a tree in polynomial time. Of course, there are non-compatible triple sets and so on. All the things are not needed here. The only thing we need to know is that there is a tree that returns a tree for a given triple set that I derived from some tree in polynomial time. Okay, so now we wanted to know what is the phylogenetic signal of this relation. And if you don't have a clue, what are you doing? You look at simplified examples. And so what we have done is we looked at all 3-leaf Fitch trees and the relations that they explain. And what you can see here is all possible rooted phylogenetic trees on 3 leaves together with all possible transfer, non-transfer edge labeling. And these trees are ordered in a way that in one line, the trees in one line, they encode the same relation on 3 vertices, right? For example, we could have a relation where we everywhere have bidirectional arcs, which means all of them are in this Fitch relation, right? X points to Y, Y points to X, and so on. So all these 3 trees explain this relation. We may have also a relation on 3 vertices that none of them, where there are no arcs, it's simply we have no transfer edges, and so on, and so on, and so on. Now there are precisely 4 trees other way around. There are precisely 4 relations that are explained by a unique phylogenetic tree. And these are the relations A5, A6, A7, and A8. And this was the first hint that we got, and it's easy to see, right? So consider now an arbitrary relation that we have obtained from some tree. Now, if this relation contains A5, for example, as an induced subgraph, what does it mean? I mean, there is only one tree on 3 leaves that can explain this relation. And this means that any tree that explains this relation must display this particular triple, in particular this edge-labeled triple. Right, we know that on the path from here to here, there must be a transfer edge. On the path from here to here must be a transfer edge. While on the remaining passes, there are no transfer edges. And this is what we call informative triples. So whenever you have a relation, a big relation, not only on 3 vertices, and you find such a subgraph, you can collect this as an informative triple that must be displayed by the tree that explains the entire relation. And the same is true for A6, the same is true for A7 and A8, and so on. So we can collect all these informative triples. We collect them in the set R star of this given relation. And I mean, for free, essentially, we get a lemma that tells us that all triples in this set R star must be displayed by any tree that explains the underlying relation. So, I mean, of course, we wanted to understand this in some more detail. Are these triples also sufficient, right? And what we needed to understand to this end is a bit more, when do we have a least resolved tree for a given Fitch relation? You can see here 3 different trees that all explain this particular relation here. Now, when is a tree least resolved? A tree is least resolved if any edge contraction in this tree, together with any relabeling, would yield a tree that does not display, that does not explain our given relation anymore. Now, we cannot contract edges and find a new labeling such that the resulting tree still explains our given relation. Now, let's have a look at non-transfer edges. Assume that we have an inner edge, and an inner edge is an edge where that is adjacent or that is incident to non-leaf vertices only. Now assume we have a non-transfer edge as inner edge. What does it mean? I mean, we ask about the relation from X to all the remaining Y in this subtree here. I mean, this non-transfer edge doesn't tell us anything. Whether X is in relation to Y or not does only depend on the particular subpath below this particular non-transfer edge. We do not learn anything about inner non-transfer edges. Yeah, so we can contract it. All inner edges in a least resolved tree that are non-transfer edges can be contracted. So all inner edges in a least resolved tree therefore must be transfer edges, right? So an example is shown here. We have here a transfer edge, and it simply doesn't matter if we contract it or not, because all the information is contained in yes, there is a transfer edge, or no, there is no transfer edge, and all these things happen below this non-transfer edge. So we can contract it and get this tree. Now, what is the second important thing we need to know is this is an edge when an edge is irrelevant. So an edge is simply irrelevant if its labeling does not affect the relation, right? So if I have an edge, an inner edge, that I can relabel to a non-transfer edge, and it still gives me the same relation, I mean, then I can also contract this edge. It does not have any information for us. Yeah. So if it is a transfer edge, I can label it to a non-transfer edge, then I already know I can contract it because I do not have inner non-transfer edges. So all inner edges in a least resolved tree are transfer edges and must be relevant. This is the conclusion. And this is essentially the characterization of least resolved trees with respect to a given Fitch relation. As we have seen before, all inner edges must be relevant, and in particular transfer edges. And this is equivalent to all inner edges are transfer edges, and there is always an outer edge. An outer edge is an edge that is incident to a leaf. That is a non-transfer edge. Right, so if I have, like in this case, only outer transfer edges, then I know it doesn't matter whether this is a transfer edge or not, because I mean, it only depends on what happens here on the leaves, on the outer edges to the leaves. So it is an irrelevant edge, and I can contract it to obtain this tree here. Now, back to our triples, what you can see is they are least resolved in a certain sense because they almost satisfy these conditions here. And I mean, except this one, but this is not so important. The important thing is that every inner edge is a transfer edge that is distinguished by a triple. This is an observation that we had. So if you look at the least resolved tree, when one can use the structural information of least resolved tree and show that for each edge, there is a particular triple, an informative triple that distinguishes this edge. And this means, yeah, that the informative triple set identifies our edge label tree T lambda, which means that any other tree that also displays this triple set must be a refinement of this tree, which essentially tells us that the least resolved tree is unique. That's yeah, the first observation that we had. Okay, so back to our question, the phylogenetic signal. Can we reconstruct the original tree for a given Fitch relation? And the answer is yes, to some extent, right? We can construct for a given relation these least resolved tree with our build algorithm replied on the informative triples. Yeah, and we know this tree is unique, and any other tree must be a refinement of this particular least resolved tree. And so this is the information that we get. We get, yeah, a least resolved version of our original tree. This is a phylogenetic signal that is contained in this particular Fitch relation. Good. Now let's go to the characterization. Now I give you an arbitrary binary relation that we may have obtained from some inference data. somehow sequence similarities. And now I ask you, does this reflect the structure of a Fitch relation? So is it a Fitch relation? And this is precisely, or if and only if there is a tree T lambda that can explain this relation. Back to our informative triples and our trees on the 3 leaves and all the possible relations on 3 vertices that can be explained. There are graphs on 3 vertices that are not reflected here. These are particular, these guys here, F1, F2, F3 to F8. All these 3 vertex subgraphs cannot be explained by any Fitch tree. There is no way, because I mean, we have here fully enumerated all the trees. This shows that informative triples are not sufficient. So if compatibility of these triple sets is not sufficient, because for these forbidden subgraphs, so to say, the triple set is empty, right? I mean, if I look at this guy here, I do not obtain a triple set because the only informative triples that I derive are from these 3 vertex subgraphs here. So we need some more information, and this is the second theorem, that a relation is a Fitch relation if and only if it does not contain one of these forbidden induced subgraphs F1 to F8. And yeah, please apologize that I do not give the proof here. The first proof that we had that was published in 2018 was an ugly, say a difficult induction proof. So it was really a quite heavy construction part, utilizing many substructures that we also observed. And it's, I mean, we do not learn much from that proof. There is an alternative proof that we found later that is based on so-called complementary neighborhoods, which is very elegant. But it also needs more information about substructures that I don't want to explain here. So just believe me that this theorem is correct. And since this is, I mean, more or less a seminar on complexity, I decided to talk more about the algorithmic aspects now, rather than to providing the proof of this theorem. Okay. I mean, what we get for free is an O of V to the power of 3 recognition algorithm, right? We simply check for all 3 vertex subgraphs whether they correspond to one of these forbidden subgraphs, and if not, we know we have a Fitch relation. Fine. But what happens now if you want to also get the tree, right? So we need to get the triples and so on and so on. And the question is, can we do, instead of using a cubic time algorithm, can we do it in linear time, the recognition as well as building the trees given the Fitch relation? And the answer is yes, and this is a part I'd like to show now. And to this end, I need a short intermezzo into so-called directed cographs, complement reducible directed graphs. So a Dico graph is a very special directed graph, and it is recursively defined. A Dico graph, a single vertex graph, the K1 is a Dico graph. And if I have 2 Dico graphs, G and H, their disjoint union is a Dico graph. Their bidirectional join, that is, I add all edges from vertices between the vertices of G and H in both directions. This is a Dico graph. And the directed join is a Dico graph, that is, I decide to take one graph G and add all the edges from G to all the vertices in H. This is a definition of a directed cograph. Now we may ask, is this graph here a Dico graph? Well, and now we can simply apply the definition. Right, we know that the single vertex graph is a Dico graph. So the single vertex graph A is a Dico graph, the single vertex graph B is a Dico graph, and so their disjoint union is based on this definition here. And the same is true for C and D, right? So C is a Dico graph, a single vertex graph. D is a single vertex graph, is a Dico graph. And so their bidirectional join is a Dico graph. Now we have two Dico graphs, and so their directed join is a Dico graph. And we see this is a Dico graph by definition. But we have learned something more. There is a hierarchical structure in there based on the operations that I have used. And the hierarchical structure defines a tree. And in fact, every Dico graph has a Unicode tree representation. And this is now, in contrast to edge-labeled trees, it is now a vertex-labeled tree. Okay, so this is a tree representation of this Dico graph. In the hierarchical structure, the clusters are given based on the operations that we had to define this Dico graph. And the last common ancestor here is 0 if A and B have no edge. The last common ancestor is a 1 if between these two vertices there is a bidirectional edge, like here. And this is an order tree, so we can distinguish between left and right. And this arrow 1 indicates that the last common ancestor is an arrow 1 if all the vertices from here point to all the vertices to here. This is simply the tree structure or the definition of the code tree of a given directed cograph. And so the recognition of Dico graphs and the construction of this code tree can be done in linear time. And there are many results established. This is all goes back to so-called modular decomposition of graphs. And so these are well-known results, so to say. And there is also a forbidden subgraph characterization of directed cographs, and the forbidden subgraphs of directed cographs are shown here. So I don't want to go too much into the details, but let's have a look at our forbidden subgraphs, right? We also had forbidden subgraphs. They are here. And if we look now, what can we see? We see F2 is a forbidden subgraph of a Dico graph. F3 is a forbidden subgraph of a Dico graph. F4, F5, F6, and so on. No, except the red one. So, but we know if we have a graph that contains this as this large one as a forbidden subgraph, if it contains this guy as an induced subgraph, sorry, then it also contains F1 as an induced subgraph, and so on and so on. And this implies that a Fitch relation is a directed cograph that in addition does not contain F1, F5, and F8, so F1, F5, and F8 as an induced subgraph. And this helps us. I mean, we know that Dico graphs can be recognized and that trees can be computed in linear time. Okay, I mean, still, we need to check for F1, F5, and F8 as an induced subgraph, right? Since even if we would have a cograph, we need to ensure that we have also a Fitch graph, which also does not contain these guys. But now we can employ the code tree structure. Assume that we have an F1 subgraph as an induced subgraph. How would the code tree look like? Now the code trees, they are shown here, for F1 induced subgraph looks like that. So locally, so to say. So we have here the vertex C, which is separated from the rest. So the last common ancestor must be a 0. And we have an edge A, which points to B. And so we have an order tree. So A is left of B, and their last common ancestor is an arrow 1. So this means we have an F1 subgraph whenever we find in the tree two vertices, where V is below W. W has as vertex label a 0, and V has a vertex label arrow 1. And now we can examine this for F5 as well. So if we have a 0 followed by a 1, then we have a F5 subgraph. And if we have an arrow 1 followed by a 1, we have an F8 subgraph. And so the lemma is, if we have a Dico graph in its co-tree, then we know that this co-graph contains one of these forbidden subgraphs whenever in the corresponding tree these situations occur. So recognition of Fitch graphs can be done in linear time. We simply build in linear time the co-trees and then check along the edges of our co-tree or the passes in our co-tree whether such labelings occur. Okay, so I mean, but now we have a co-tree that is a vertex label tree. We also want to have a Fitch tree, right, which is an edge color tree. And this can be done in a very simple way. Whenever you observe a 0, it means that they are not in this Fitch relation. That is, along their passes, there is no horizontal gene transfer at all. You simply put these edges here as non-transfer edges. When you have a 1, then you simply know that D points to E and E points to D in this Fitch relation. And so you simply add these transfer edges here. And for an arrow 1, you replace this by a caterpillar. So you can see it better on this example. So an arrow 1 means, and we have an ordered tree, that x1 points to x2, x2 points to x3, and so on. x2 does not point to x1, but it points to x3 and all the remaining ones. And so we can simply replace it by such a caterpillar in a way that we here along the backbone, we have transfer edges, and we simply add it here. And this can also be done in linear time. So essentially we get a Fitch tree in linear time as well. And this is a theorem. Whether a relation is a Fitch relation or not, this can be done in linear time, and its unique least resolved tree can be computed in linear time then as well. Good. I started a bit late, so I still have a few minutes, right? Yeah, okay. Back to the original definition. I mean, this was a symmetrized version that is on the path between x and y, there is a transfer edge. This was the original version of Walter Fitch. We have here considered a path from the last common ancestor to y, but the symmetrized version is now quite easy to understand because it only matters whether on the path there was a transfer edge or not. So if we have this scenario here, we would get simply an undirected edge. And in case we have here a scenario like that, we get an undirected edge, and the same is true here. And here we get no edge at all. So we simply lose all information about directions. That's all what happens. And I mean, this already shows that the least resolved trees are not unique anymore, at least in their labeling, because this tree would explain the same edge as this tree here between x and y. But anyways, if you look now to our allowed and forbidden subgraphs, and we simply ignore the directions, and we still know that we can explain this graph here by a Fitch tree, namely precisely the one, for example, that explains this relation here. But then this F1, this F2 subgraph, after ignoring the directions, is not a forbidden subgraph anymore. Okay, and the same is true for the other ones. The only remaining subgraph that remain is an edge with an isolated vertex. Whenever you have a 3-vertex and 2 subgraph that is isomorphic to an edge and an isolated vertex, then you know it's not a symmetrized Fitch relation. And for those that are very familiar with the topic is, I mean, what is the characterization of complete multipartite graphs? It's precisely the fact that they do not contain an edge with an isolated vertex as in Q subgraph. And so a symmetric Fitch relation must always be a complete multipartite graph. This is the characterization. You can see here an example of a Fitch tree that explains this complete multipartite graph. We have here 1, 2, 3. They form an independent set. We simply add them with non-transfer edges, and 4 and 5 as well. And then we connect all these guys with transfer edges, and this is it. So one has to say what this shows, that this is symmetrized Fitch relation does not contain much information about phylogenetic histories at all. Okay, so let's summarize here. So what are the messages? It is first the Fitch relations, the directed version. They have a uniquely determined least resolved Fitch tree. This was based on the informative triples. Any Fitch tree that explains a relation must be a refinement of the least resolved tree. Fitch relations, these directed versions, are characterized by a small set of forbidden subgraphs on 3 vertices, and they form a subclass of directed cographs, and the recognition of these relations can be done in linear time. And the class of symmetrized Fitch relations is simply the class of complete multipartite graphs. Just a bit outlook. I mean, we can generalize now. This is now a nice mathematical playground, right? Instead of using a 0, 1 label, I mean, why not using a 0, 1, 2, 3, 4, 5, 6, 7, 8 label, right? And look what happens now. Now you get a bunch of, so to say, a multi-graph. And the question is now, can I find an edge label tree that still explains me this graph here? It's a nice mathematical playground, and yeah, I like it very much. A further result I need to mention is what we have. I mean, we have just looked on pure 0, 1 edge label trees, transfer, non-transfer edges. What we have not taken into account so far is, are all these trees with 0, 1 edge labels also biologically feasible, right? So assume that we have this Fitch tree here, and A prime are from some species A, and B prime are from some species B. Now the species tree would look like that. Since there is no transfer edge on the path between A and B, there is no way I mean, I can only place this part onto the species tree in a way that the last common ancestor of A and B is here or above. And the same is true for A prime and B prime. But then transfer edges must be mapped incomparable into the species tree so that they can transfer material across distinct species. But this is not possible because all the transfer edges are then placed here. So this would not biologically feasible, so to say. And the characterization is still open of such biologically feasible Fitch trees. So some open problems. We want to know more about Fitch trees, in particular also later when we know about biologically feasibility. We have to take into account time consistency because we don't want to have back edges or go into the past when we reconcile these trees. So in fact, the question is how much information about the species tree is contained in such Fitch relations. Still an open question. What we also don't know in the moment is what is the complexity of editing an arbitrary relation to a Fitch relation. So it's unknown whether it's an NP-hard or not. I suppose it's NP-hard, but I don't know. The question is also how easy gets a problem when we already have a Dico graph. It's easier to just remove F1, F5, F8 forbidden subgraphs from a given Dico graph? Gets a problem then easier? I don't know in the moment. And of course, one can generalize this. I mean, maybe we do not ask for a tree, but ask if it cannot be explained by a tree, is there a 0, 1, a non-transfer, transfer edge labeled network that can explain it? Maybe this is of interest as well. And of course, inference, inference, and inference. And this is one of the big building grounds that we need to understand is on how to infer these relations in a nice way. So there are several people that are involved in this project on Fitch relations. That is in particular Manuela, Peter Stadler, Nicolas Wiesecke, John Anders, Carsten Seemann, Zhangjing Long from China, David Schaller, and Manuela Fong. And so I'd like to thank you, and that's